The structure and function of neurons. There are 100 billion neurons in the human nervous system, 80% of which are located in the brain. By transmitting signals electrically and chemically, these neurons provide the nervous system with its primary means of communication. There are three types of neurons: motor neurons, sensory neurons, and relay neurons. The features of each are illustrated in the diagram. The structure of neuron. Neurons vary in size from less than a metre to up to a metre long, but all share the same basic structure. The cell body, or soma, includes a nucleus, which contains the genetic material of the cell. Branch-like structures called dendrites protrude from the cell body. These carry nerve impulses from neighbouring regions towards the cell body. The axon carries the impulses away from the cell body down the length of the neuron. The axon is covered in a fatty layer of myelin sheath that protects the axon and speeds up electrical transmission of the impulse. If the myelin sheath was continuous, this would have the reverse effect and slow down the electrical impulse. Thus, the myelin sheath is segmented by gaps called nodes of Ranvier. These speed up the transmission of the impulse by forcing it to jump across gaps along the axon. Finally, at the end of the axon are terminal buttons that communicate with the next neuron in the chain across a gap known as the synapse. When a neuron is in a resting state, the inside of the cell is negatively charged compared to the outside. When a neuron is activated by a stimulus, the inside of the cell becomes positively charged for a split second, causing an action potential to occur. This creates an electrical impulse that travels down the axon towards the end of the neuron. Synaptic transmission. Neurons communicate with each other within groups known as neural networks. Each neuron is separated from the next by a tiny gap called the synapse. Signals within the neurons are transmitted electrically, however signals between the neurons are transmitted chemically across the synapse. When the electrical impulse reaches the end of the neuron, the presynaptic terminal, it triggers the release of neurotransmitter from tiny sacs called synaptic vesicles. Neurotransmitters are chemicals that diffuse across the synapse to the next neuron in the chain. Once the neurotransmitter crosses the gap, it is taken up by the postsynaptic receptor site, in other words, the dendrites of the next neuron. Here the chemical message is converted back into electrical impulse and the process of transmission begins again in this other neuron. Several dozen types of neurotransmitter have been identified in the brain, as well as the spinal cord and in some glands. Each neurotransmitter has its own specific molecular structure that fits perfectly into a postsynaptic receptor site, similar to a lock and a key. Neurotransmitters also have specialist functions. For instance, ACH is found at each point where a motor neuron meets a muscle, and upon its release will cause muscle to contract. Neurotransmitters have either an excitatory or an inhibitory effect on the neighbouring neuron. For instance, the neurotransmitter serotonin causes inhibition of the receiving neuron, resulting in the neuron becoming more negatively charged and less likely to fire. In contrast, adrenaline, an element of the stress response, which is both a hormone and a neurotransmitter, causes excitation of the postsynaptic neuron by increasing its